Last time we talked about prokaryotic and eukaryotic, did we?
Yes, we did.
Yeah, yes, we did. Okay, good. Okay, what we can see here, what did I say about the prefix pro when you see pro? What did I say? Because I know you have a lot of okay, but there is something that I said about pro.
It comes
Primitive.
before
Primitive.
primitive.
Yeah, I did say primitive, right? Yeah. That means like those organisms that are prokaryotic, most likely they have a primitive nucleus. And we call that nucleus primitive. Uh, we use this vocabulary just to make people understand that it's not that they do not have a nucleus, but that nucleus is not fully formed. Therefore, it's not delimited by some kind of a nuclear membrane. Therefore, we say that they are prokaryotic. Yeah, because it's not fully formed. And I did talk about also nucleoid. What did I say about nucleoid? Because they have a nucleoid region.
It's a false nucleus.
Thank you very much. They have this kind of yes false nucleus. Okay, but it contains DNA, as we can see here, contain also RNA. Then in prokaryotic organisms, we are going to see that some prokaryotic organisms they have another type of DNA. Yeah, that they call an extra chromosomal DNA. Now, bacteria specifically, bacteria originally didn't have, yeah, an extra chromosomal DNA. That extra chromosomal DNA was acquired due to okay due to adaptation, due to evolution. Okay, that means those organisms, these kind of organisms, most likely because they were to adapt to their environment, to certain conditions. Therefore, they have developed, okay, an extra chromosomal organelle or apparatus, not really developed, but something like that they pick up, you understand? They can pick them up, for example, from other bacteria, okay, other organisms that they would pick them up, okay, and those extra chromosomal DNA, they would contain information for them to be able to adapt to their new environment, to resist certain things that are deleterious, to them. Okay, that therefore you may see some prokaryotic organisms, they may have a plasmid. Okay, normally eukaryotic DNA, if you remember, very elongated, okay, very elongated, the eukaryotic DNA, okay. In fact, because it's very elongated, for that DNA to fit into that cell, that eukaryotic cell, therefore you have to wrap, yeah, that DNA around an apparatus, around some kind of proteins. And if you remember, we call them histones, those histones, okay? And if you remember also, when you have like 6 to 7 to 8 histones, of course, they are going to form what you call a nucleosome. They form a nucleosome. And of course, that nucleosome is going to be part of that material that you call chromatin. And of course, the assemblage of all of those chromatin would form what you call your chromatid. And of course, this is chromatid, they are going to be your chromosomes. Now, microbial cells, most likely in nature, you are going to see them and they are going to live specifically as colonies. Yeah. Okay. They are going to be a community of microbes, most likely. Yeah. And of course, why? Because they will put themselves together so that they can survive. Okay. Most likely, they are going to use their nutrients and they are going to use certain enzymes, and those enzymes are going to be tools uh, for them, okay, to catalyze reactions, and reactions that are anabolic and reactions that are catabolic, okay, therefore reactions that are part of their metabolism, okay. You are welcome to read this. Unfortunately, I cannot because it's very, 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 very tiny to me. But understand that some cells they have their properties, but all cells, most likely, you understand they are going to undergo some types of metabolism. They would go or develop, and of course, they would evolve. But some cells can differentiate themselves into some kind of other structures. For example, some bacteria, when they are under some kind of dire conditions, one of the things that they are going to do, yeah, they are going to undergo sporulation. Therefore, now, medically important bacteria, I need the fullest of your attention. The fullest of your attention that I need. Please write for me. Bacillus. Let me see if I can write it down. Bacillus, medically important bacteria. Wait for me just a minute, please. I'm on the chat. In the chat. Okay. We are talking about the medically important bacteria. Medically important. Come on, I'm typing it, it's not responding. Wow. Okay, let me see. And Clostridium. Okay. In the course of your practice, when you hear, okay, the word bacillus, for example, Bacillus subtilis, Bacillus megatorium, and not megatorium, but subtilis, Bacillus cereus, for example. You hear Bacillus as, the genus, okay, Bacillus. And you hear also the name Clostridium, C-L-O-S-T, Clostridium, okay, like the Old English, to close, or cloistered, close, C-L-O-S-E, okay, Clostridium. When you hear, okay, those genera, they are part of the very important bacteria. Why are they important? Let me write it down for you. Sporulation, that we will talk about, okay, especially in my lab. We'll talk about sporulation. Okay? Because they will undergo sporulation so that they can protect themselves against an adverse condition. Those are also those kind of gram-positive bacteria that's going to make okay, your patient's life miserable. That's all we are going to say right now. Okay? Yeah. Once you see bacillus, you hear bacillus. You hear what? Clostridium tetani, Clostridium botulinum. Okay? Think about sporulation. Very, very important. Let's continue. Now, some microbial cells, of course, they are motile, and of course, they are going to use some kind of an engine. They are going to use some kind of an, we can see that some kind of appendages so that they can actually move. Okay? Yeah. Some of them can use a flagellum, can use flagella, can use also cilia to actually to move. They can use some axial or axial filaments to actually to move. Very, very, very important. Yeah, very, very, very important. Now, because they would self propel themselves, using some kind of apparatus. Okay. Now, some of them can actually, at some point in time, they have an appendage, they have some kind of a structure, and they are going to modify it so that, they can become some types of specialized cell. Now, microbes can actually, uh, communicate with each other. They can use their old phone lines to actually to call each other. Okay, kind of like not too recently, but it's not really too long ago. What they discovered that microbes, especially bacteria, prokaryotic organisms, they use something that we call nanowires. Therefore, the same way, for example, that you have your own phone line, they would have some lines where they would attach to each other so that they can communicate via nanowires. Okay? Now, in terms of activity, of course, yes, organisms can evolve, and we understand the key word about evolution is going to be adaptation. Okay? And of course, I don't know if I did say that in my lab. Okay? Yes, evolution, of course, adaptation, the organisms can actually mutate themselves so that they can adapt to some kind of an environment. Yeah. But it's not all the times that some kind of a mutation to adapt to an environment is good for an organism, especially, for example, for us in the context of, for example, malaria and sickle cell anemia, for example, right? Okay. Yeah. Because we, our body to fight against malaria, okay, the cells could become sickled, but at the same time, it seems like it's better to have that malaria and deal with it than having sickle cell anemia. Therefore, the Earth has been here, okay, for 4.5, 4.6 billion years. Okay, and of course, the Earth, therefore, the first cell would appear between 4 to 4.3. And of course, at the beginning, the atmosphere was anoxic. There was no oxygen. Okay? Yeah. Therefore, the metabolism, when we're talking about metabolism, it was anaerobic. Okay, I'm sure you had plenty of anaerobic metabolism in your biochem class. Okay, it was anaerobic. Okay, then we started to have phototrophic organisms, and because of that phototrophic organisms, most likely they would use the energy, from the sun, okay, so that they can fix carbon dioxide with water, yes, to produce some kind of food for us, and of course, to produce oxygen, you understand, and now because they are producing those oxygen, those phototrophic organisms, and of course, the earth now is was becoming oxygenic. Okay, therefore that's why plants and animals most likely, that are like heterotrophic, that are also, you know, iophile, most likely those organisms, they will appear later on, because when the oxygen is basically, okay, available. 1976-79, if you remember, okay, we had the Carolus Linnaeus, Carl von Linné classification, those KP, stuff, those kingdom, phylum, those, okay, family, class, order, you understand, genus, species, okay? That was the classification that we had. However, if you remember from some of your previous classes, ribosomes, they are made of two subunits, a large subunit and a what? And a small subunit, if you remember. And if you remember, in the eukaryotic organisms, ribosomes, they would come from the nucleolus. You have to specify from the nucleolus. And what are ribosomes? Ribosomes are mostly RNA. That means that ribonucleic acid mixed with some kind of protein. And of course, the large ribosome and the large subunits and the small subunits, they come from the nucleolus. And if you remember as well that they are not, okay, fully functional. They are going to be functional provided that that large subunit and that small subunit, they are going to meet together and they form a ribosome. Then now, okay, that entire unit is going to be fully functional in the cytoplasm. Therefore, if we say that ribosomes are made of RNA and what protein, therefore we understand that there is a sequence for the large subunit and there is also a sequence for the small subunit. And because we say so, that means scientists that have been analyzing the sequence of the large subunits and the small subunits, they have discovered something. What have they discovered? Well, as much as I said to you, I know that I said that, the first time that I was exposed to microbiology, I was 14 years old in Haiti, some 44 years ago. And I don't know if it's in the lab, okay, but I said, well, we have bacteria, we have the archaea bacteria. That's what we used to call the archaea bacteria. Okay, those organisms that can actually live in extreme environment, the archaea bacteria. But later on, they had to remove the name bacteria with the archaea. Why? It's because when they analyzed, okay, the RNA of the small subunit, when they analyze the RNA of their small subunit, what have they discovered? They have discovered as much as archaea are unicellular, like a bacteria, as much as, they lack organelle. as much as they are prokaryotic because they have some nucleoid region, they don't have any form, they have a primitive nucleus. Okay? They undergo binary fission in general. Yeah. Of course, yeah, they have something that looks like a peptidoglycan in the cell wall, like a bacterium. It's not peptidoglycan, it is pseudomurine that we are going to see. It just happens that the sequence of the RNA sequence of the small subunit, that RNA sequence has more similitude, has more similarities to the RNA sequence of eukaryotic organisms than prokaryotic organisms. And they are like, aha! Therefore, as much as the archaebacteria, they are prokaryotic, but they are not bacteria. Therefore, we need to remove that name, okay, archaebacteria. We need to remove bacteria from their name. Therefore, they are going to be in a class by themselves. Let's say they are going to be in a domain by themselves that we call archaea. Therefore, George Fox and Carl Woese, W-O-E-S-E, they actually divided, okay, yeah, organisms into 3 domains. Okay, of course, we have the prokarya and we have the eukarya, but the prokarya, what do they have? They have bacteria, they have archaea, that's number 2, and everything else is eukarya. Be very careful. When we say everything else is eukarya, you need to make abstraction of viruses and prions, because prions are not, okay, microorganisms. Viruses, when we will be doing those viruses, some of you may agree, some of you may not agree, okay, that viruses are not really microorganisms, okay, in their own right. We will talk about that at a certain time. And when they were doing some more research, what have they discovered? They discovered that, organisms are descended from the last universal common ancestor. Some microbiologists, the looker is really the archaea. Okay? You would read or consult some microbiology book whereby they would say, of course, there is bacteria, but the looker, that means everything else come from the archaea. Yeah, that means the real looker, the last universal common ancestor, yeah, okay, was really the archaea. Therefore, archaea can be extremophile because they can actually live in habitat, okay, that may be very harsh for other life forms. For example, in hot springs, in glaciers, okay, high salt content, very, very, very important. High acid or alkaline content that can destroy the DNA, can destroy protein, zones of high pressure, very high pressure, all the way down, okay? For example, into the Marianas, for example. The extremophile or extreme thermophiles sometimes, in very hot vents, and you will find some organisms. High salt content, because okay, that, if an organism lives in a high salt and salty environment, okay, because you understand that salt can actually get into the organism, calling water, NaCl, and of course, yeah, that water is going to make that organism to expand until it bursts and it can kill that organism. I don't know if it's in my lab, that I ask some people, why do you sweat? Yes, you sweat for 2 things. Yeah. One is the physics. Yeah. You are hot, very hot, and of course, you understand now, okay, someone would sweat. You understand? Or removing, some of the high temperature, the hot temperature into your body, of course, for evaporation. But you sweat also because your sweat contains some salt. And what happens? You have on top of your skin a bunch of bacteria that can actually take that sweat and also take the water. Therefore, sweat also has some kind of antimicrobial properties because of the salty environment. When we talk about ecosystem, it's going to be all living organisms, you know, yeah, okay, that are living in an environment, yeah, using their physical and their chemical properties. They are going to metabolize because there's going to be reaction that are aimed to build something, and there's going to be reactions that are aimed, most likely, yeah, okay, to degrade something so that they can use the energy, yeah, store it, yeah, use it. So whatever that is not being used, it's going to be stored so that can be used later on. Therefore, from your biochemical perspective, most likely it's kata for ana. There's going to be catabolism, yeah, okay, for anabolism later on. That's mostly what's going on from your biochem. You know about, taking your glucose and all of those things, you are going to have, the degradation of glucose, yeah, most likely. Okay, therefore, we are going to break all those covalent bonds, releasing the energies and the electrons. And of course, you're doing something good for your body, and at the same time, it's also doing something very dangerous for your body that can kill you. That's from your biochem. Microbial ecology, yeah, is really studying some kind of organisms, most likely in their natural environment, and how most likely, yeah, the rapport, the relationship, how they relate to their environment. I'm so sorry, people. I cannot see. Oh, okay. Yeah. Now, that means when we're talking about extremophiles, most likely, it may be that there are some organisms that can be hyperthermophile. For example, the methanopyrus, okay, they are okay, and most likely they can live in a temperature that is like minimally 90 degrees centigrade. Oops. Okay. Thanks God, we haven't found any algae, yeah, that is pathogenic to humans. Okay, and I'm praying that an algae most likely doesn't. Okay, that we would find one, because if an algae would be pathogenic to humans, understand that perhaps your immune system, using, for example, the pyrogenic toxins, may not be able to do anything for them. You have the sarcophile, most likely, those are the ones, that may exist in your fridge, for example. Yeah. For example, the Psychomonas, the bacteria, you can see them in sea ice. Yeah. Of course, you have the Picophilus, those are like the acidophile, the alkalinophile. Yes, most likely you have the Nationobacterium. Okay, you have the ones, those are like the baophile or the baophile, the Moritella, for pressure, and you have the halophile. Most likely those are the ones that can really live in salty environment. Microorganisms can be both beneficial and harmful to humans. Yeah, I'm sure that you know about organisms that live, okay, in your gut. Okay, those they are part of your microbiota. Please do not say, please and please and please do not write normal flora. I know. Yeah, you have heard people saying normal flora. We don't use that term anymore in microbiology. Yeah, microbiota. Okay. Now, I'm sure you know that you have some bacteria that live in our large intestines, and they are responsible for water reabsorption. And you understand that yes, you need water in your body, you need a certain pressure that can bring that oxygen and that glucose all the way to your brain, because your brain most likely is located, okay, against gravity. You need a pump, but you need also something that can have some kind of inherent pressure, and that would be those, aqueous, you understand, this fluid, most likely water. Therefore, those bacteria, most likely, we are alive, okay, because of those organisms that are part of our microbiota, especially those ones, they are involved even, in vitamin K metabolism and production for us. They have also discovered that there are some of them that would actually fight against other bacteria. Okay? Of course, we understand, yeah, because they are territorial. You understand, therefore, they don't want any other bacteria or any other bacterium or classes of bacteria to come and invade their territory. Therefore, they are going to fight, which is good for us because they are part also of our microbiota. They are not harmful, they are harmless. Okay? And if we remember also, we have this kind of relationship between two organisms, and most of them are commensalistic. If you remember, okay, they are not, parasites. Yes, you have parasites, but some of them are commensalistic. Not some of them, most of them, they are commensalistic. That is why, in the course of you that are going to be prescribing, you need to be very careful of what type of antibiotic that you're going to prescribe to your patient, because those antibiotics can actually destroy your patient's, okay, microbiota. And now those that were harmless to you, they can turn against you. Or if they don't turn against you because they were defending your body against certain other harmful bacteria, now you're killing them. Now those harmful bacteria, they are moving in. Okay? And of course, there is a disease. Does anyone know the disease that I'm talking about? The name of the disease?
Could you repeat that? You said that the bacteria are turning against you?
Because you're destroying them. Because you destroy them by giving some kind of antibiotic. Write it down, please. That's pseudomembranous colitis. You may be giving antibiotic to someone, you understand, and then, yes, yes, you are destroying bacteria, but you may be destroying also some of the bacteria or most of the bacteria that are part of your microbiota. And because of that, okay, they cannot do water reabsorption for you. And because they cannot do that, okay, let's say water absorption, they cannot do that. Now your stool is going to be full of water, therefore you are going to have a diarrhea. Therefore, if you have a patient that you're giving some antibiotics, and then now your patient, after the course of antibiotic, or while your patient is on the course of that antibiotic, is developing diarrhea, it may be that you are hurting instead of helping. You may have to stop that antibiotic. You understand? You have to stop it, because if you do not, your patient may develop pseudomembranous colitis. Can you see the PowerPoint? Can you see the PowerPoint? Hello? Hello?
Yes.
Hello?
Yes, Professor, we see it.
Thank you very much. I have a very small computer. Yeah, thank you. Okay, therefore, microorganisms, of course, understand that they can be pathologic to humans. Okay, you know, today, well, no, we have to change it. Heart disease, yes and yes, but COVID and this, therefore, this is old. Microorganisms can be used in agriculture, in human nutrition. Yeah, for example, the beautiful jean that, pants that you are wearing, yeah, it may be that it was blanched out with E. coli. The production of indigo, for example, if you go into the certain part of France, you want to eat, for example, that cheese that people would qualify as the best cheese in the world, that you call the camembert, most likely, okay, that cheese really was being enhanced by some kind of microorganisms. If you remember, because I know this is in your generation, we had the oil spill, okay, in the Gulf, okay. Right now, they are still using some bacteria to actually to clean the spill. Not in your generation, but some of you may remember the Aquilaro, for example, in Alaska, the Alaska oil spill. Okay, if you remember that, also they are using bacteria, for example, to degrade, okay, all of those waste, because that was an ecological disaster. Yeah, and therefore bacteria can be also beneficial, okay, for us or to us. In fact, we can use bacteria, for example, to make antibiotics. We can use microorganisms to make antibiotics. Now, some bacteria, they are nitrogen fixing bacteria. We will talk about them. Okay. Some of them can degrade, for example, cellulose, and of course, some of them are part of our microbiome or microbiota. Later on, I don't know if we did that in biochem, but we will do a little bit of anaerobic metabolism, whereby some bacteria, they will have some kind of an enzyme that we call nitrate reductase and nitrite reductase that they would use most likely, yeah, so that they can reduce, okay, reduce nitrite, okay, to make to fix nitrogen for us, or to make molecular nitrogen, and of course water. Okay, some of you might have done that in biochem, but we are going to redo it here. Of course, you understand that some microorganisms, they can spoil our food, our food poisoning. Okay, most likely we need to understand that because of their impact into the human society, we have developed some food safety mechanism and protocols, okay, so that we do not get that poisoning. Very, very, very important. Okay. Therefore, that's basically your biochem in an anaerobic configuration. Understand that we can make alcohol. And today in my lab, okay, well, in my last lab, anyway, we Tuesday and Thursday, we talk about Louis Pasteur, okay? And I didn't talk too much, but the only thing that I said, when you are handling your tube, you need to keep your tube slanted. Why? To avoid contamination, okay? Therefore, if you remember, okay, from glucose would go into ethanol, and from ethanol, most likely, yeah, we can form acetic acid, that would be vinegar. Therefore, if you remember, they call Pasteur. Why do they call Pasteur? They call Pasteur, and they said, well, because Louis Pasteur was involved into crystallography as a chemist. Louis Pasteur was not a microbiologist, okay? But he discovered, okay, if there is a way for me to apply certain heat to the wine, because the wine, those people that were involved into the wine business, most likely the wine were being contaminated by some kind of bacteria, some kind of yeast as well, and was turning, okay, the alcohol, the wine, into acetic acid, into vinegar. Okay, therefore they were losing a lot of money. And because of that, they called Louis Pasteur. Louis Pasteur discovered that if I apply a certain heat, okay, most likely it's going to destroy the organism, those microorganisms, but it's not going to spoil the taste of the wine. Okay, and that's what, of course, we call it pasteurization. Okay, in our body, if you remember again from your biochem, the glucose would turn itself into pyruvic acid, and of course, in an anaerobic environment, yes, okay, the pyruvic acid is going to turn into the lactic acid. And thanks God that, we have a Cori cycle. Okay, some of you remember the Cori cycle, whereby this lactic acid, most likely, yes, they are going to turn into glucose as well. This is kind of like gluconeogenesis 2.0. Yeah, okay, so that we can continue to live, because most likely if we were to have an accumulation of lactic acid in our muscles, and if it wasn't for the Cori cycle, we won't be able to actually to move. Most likely, what time is it, please?
7:20.
7:20.
7:20. Okay, good. Because I'm going to take just a 5-minute break. Remember, okay, and then we will continue. But let's continue here. Let's finish with this. Okay. Now, biofilms. Let me ask you, did I talk about quorum sensing the last time? Last Thursday, did I say anything about quorum sensing?
Yes, you did.
Yes, I did. Thank you very much. Yes, thank you. And if you remember, what did I say about quorum sensing? If you remember. Anyone, please.
I have that it forms colonies and communities, and when one species comes to a place, it acclimates itself and allows other species to come and create a community.
Yes. And you're also right. Yes, biofilm. Yeah. Okay. Yeah. We are going to see biofilm on some submerged surfaces. Okay. Medical devices in tanks. You understand, you have biofilm, for example, on top of your teeth right now, because if you pass your tongue on top of your teeth right now, okay, on the surface of your teeth, you can feel that slippery stuff. Those are biofilm. Okay, we talked about quorum sensing last time. Okay, caused by quorum sensing. Well, they can use microbiology in the industry, you understand, to make some kind of, products, antibiotic, producing enzymes, some chemicals, some hormones, okay? Biotechnology. Now, very, 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 very important. When you see that word biotechnology, okay? There is another word that is closely related to it. Even though that I took the MCAT, I believe that was in 1985 or something like that. I remember vividly. There was some kind of a question, some biology, excerpt that I was reading in my MCAT, and it's the word specificity, specific biotechnology. The word specificity, it has to be specific, people. Because in biotechnology, let's say you are doing DNA recombinant technology. I know that some of you in biotechnology, you have done some with Dr. Williams and stuff, you know. Well, if you don't know what you're doing, I ask you specifically, what are you trying to achieve, and you don't know, you might end up having something very undesirable that you don't even know how to control. That's why in biotechnology, you have to be specific. You need to know what you're doing. You need to know what you are looking for. Very, very important. In fact, you have seen that there are some application of biotechnology that end up with something very undesirable. Okay? Productions of biofuels, methane, ethanol, for example, in Brazil, yeah, they are using corn, most likely to produce some kind of a petroleum. Okay? Wastewater treatment, definitely. Bioremediation using bacteria. Okay, that's going to clean, separate all of those, you understand, undesirable products. Okay? Very, very, very important. To clean up pollutants, we already talked about that. And now, you know about microscopes. We talked about it. If you remember Anton or Antony van Leeuwenhoek, who was like a Dutch, was a haberdasher that I talked about in my, some kind of like Victoria's Secret for men. Okay, most likely Anton van Leeuwenhoek was, his hobby was to play with a bunch of lenses. And that's why they say that he's the one that was using, okay, most likely the first microscope. Okay, of course, you understand that the Chinese, they had someone from China that discovered it, and you understand also that the Russians, after 1917, the Bolshevik Revolution, they had to throw away a bunch of science book, and then everybody that would discover everything in the world, they were Russian, they were not anyone else. Therefore, the people, you understand that, okay? Therefore, he is the one that first described bacteria. In fact, he described also some organisms that he saw that were alive, and he called them animalcules. Robert Hooke was the first to describe microbes, but Robert Hooke, if you remember, was the first one that coined the term cell, C-E-L-L. Why? Because he saw structures that look like a cell in a gel. Therefore, that would be the microscope used by that first one by Robert Hooke, and that smaller one, that was by Anton van Leeuwenhoek. Now, therefore, we talk about magnification, the ability to enlarge something, and resolution, that would be the ability of an optical device to distinguish the distance between 2 points in space. Okay? But understand that, okay, our eyes are limited in terms of resolution, which means, like, for example, if an object is kind of like, less than 0.2 millimeter, we might not be able to distinguish the distance between 2 points in that object. Therefore, if there are two objects and they are tinier than, that measurement, it may be that you would see them as one. We cannot distinguish them. Light microscope, as much as it can magnify an object all the way to 1000x, therefore we understand that the limit of resolution for a light microscope is about 0.2 micron, which is 0.2 times 10 to the power of minus 6 meter. Now, in terms of microscopy, we kind of have like 7 microscopes that would take into account the properties of light. Okay? I didn't say light microscope. I say 7 microscopes that could take into account the properties of light. Of course, in terms of light microscopy, yeah, we can have 5, but there are 7 that could take into account the properties of light. Okay? We have bright field. Most likely, this is the one that we use in the lab at the North Campus. There is phase contrast that we will talk about, phase contrast. We have GIC, which is also phase contrast, but using kind of like 2 beams of light so that you can have a better, 3D resolved image. Dark field, of course, it tells you that the field is dark, therefore the object that you are looking at is going to be brighter than the field, as opposed to bright field. We have fluorescence microscopy. We use that same technique also in cancer research for fluorescence microscopy that we will talk about. Therefore, that would be a light microscope. Of course, you can see the ocular lenses that we call the eyepiece. Okay, you see the objective lenses, and you could place your specimen in the slide. You have the diaphragm that will contain the condenser lens and the opening with the iris. And of course, you have the illuminator that you call the lamp. Therefore, what is a compound light microscope? Well, a compound light microscope most likely would use visible light, okay, so that you can illuminate a cell. Therefore, there would be some kind of 2 sets of lenses for the image. You would have the objective lenses, yeah, and you have the ocular lenses. Most let's say, middle price microscope that we have at colleges and basic microscope that are compound light, most likely the ocular lens is going to be 10x, and of course you would have like 3 to 4 objective lenses. You have the 4x, the 10x, the 40x, and of course you have that 100x. Therefore, the total magnification is going to be the magnification of the ocular lens, lenses, okay, or lens, that depends if it is inocular or binocular, okay, multiplied by the objective magnification. Yeah, therefore, could be 10 times whatever the objective lens magnification is. But since it could be 10 to 30x, therefore, if the magnification of the ocular lenses by themselves is 30x, therefore it's going to be 30x multiplied by the magnification of the objective lenses. Therefore, that's how we would calculate the total magnification. Therefore, for most light microscopes, okay, a magnification of 1000x, that's what is required so that we can have, okay, the highest resolution that it can achieve, which is 0.2, okay, micron. In bright field illumination, the specimen is visualized, okay, because there is a problem with the density, not a problem, because there is a difference between, the density of the specimen, the specimen and its surroundings, okay? In the lab, we talk about the 3 attributes of a light microscope. We said magnification, we said resolution, and we said also contrast. And we talk about contrast in the context of refraction. We covered refraction kind of like in the lab, whereby, okay, light, it's going to bend, and then when it hits an object, it can refraction or refractionate it, can make it look bigger or smaller. That depends on, okay, the difference in index of refraction or refractive index of the two milieu. That depends also on the lenses that are used. Okay, those of you that have done physics optics, you know the difference between a convergent lens and a divergent lens. I'm not here, I don't think that I have the intellectual acumen to teach optics. Now, therefore, you look at your microscope, okay, therefore you can see the light path, yeah, okay, and how the image is being visualized, and you have your eyes. Today, in the lab, I talk about decussation. We talk about the optic chiasma. We talk about the sphenoid bone. We talk about the celatostheca. We talk about the pituitary gland. We talk about ipsilateral and contralateral. Today, we talked about that. Therefore, you can see, for example, the decussation, the crossing. Yes, you can see the crossing. Here, I don't know if you can see my cursor, the arrow, the kind of like orange yellow arrow. That's the crossing, the crossing, excuse me, that we call the decussation. This is an example of bright field, because you can see that the field is bright and the object is has a different, you know, color to the field. But then, when you look at an object in a microscope, okay, I'm going, you know what, let's take 5 minutes, please. Let's take 5 minutes and let's come back in 5. Okay, can you hear me? Hello?
Yeah.
Yes,
You know,
Professor.
let's take 5, just a 5, okay? Okay. Now, when we talk about uh, visualizing and seeing an organisms under the microscope, most likely, when you take your slide, you make your smear, and then you want to see the organism, most likely you will not see anything. Most organisms, especially for bacteria, you will not see them. Well, you can see perhaps, Serratia marcescens that we have used in the lab, because at a certain temperature, that Serratia marcescens is going to give you that red color. Okay, that red color, red, the color of planet Mars, Serratia marcescens. Okay, yeah, because it has any special enzymes, okay, that you can go and find out. Okay, it has a special enzyme. It's going to turn red. Pseudomonas aeruginosa can give you that greenish color. In fact, sometimes you look, you actually do your inoculation, you incubate your organisms. When you look at the plate, you can see all of those greenish materials, and that may be Pseudomonas aeruginosa that you can actually like, oh, that's greenish, that look like Pseudomonas aeruginosa. It may be Pseudomonas aeruginosa because it's going to be, green. But most bacteria, you won't see anything. Because of that, you have to stain them. You have to dye them. That's what you have to do. Why? Because staining the organism is going to improve the contrast. That means it's going to accentuate, okay, the difference in refractive index. Because of that, you would be able to distinguish certain characteristics of that organism. Therefore, what are dyes? That's our organic compound that's going to bind to some specific materials inside of a cell, and because of that, they will give you some kind of a different color, okay, that can show certain characteristics, certain structures, okay, and because of that, you will be able to distinguish a bacterium from another one based on the color, based on the characteristics, based on the organelle, but based on the presence or absence of certain organelle, certain structures. Dye can be basic, okay? The basic are positively charged. Therefore, they are going to bind strongly, okay, to a negatively charged components of the cell. If you remember, you can have a cell. The cell has a component. That component is most likely producing a lot of ATP. The phosphate that's going to make that cell, the inside of the cell, to be negative. Cell and molecular biology, especially near, okay, the membrane of that cell. Therefore, if you want a dye to penetrate inside of a cell, therefore you may want to use a positive dye, a positive stain, because that positive stain is going to be attracted to the negative components inside of that cell. Therefore, that stain is going to bind to those components, and you'll be able to distinguish them. However, and I hope that you are listening, one of the characteristics of a cell, that is where you need to use negative staining, is because that cell is very, 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 very tiny. And because it is very, 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 very tiny, even though you use a positive cell, you would still not be able to see it because it is too tiny. Therefore, you have to see it in an indirect way. How are you going to do that? Well, we are going to stain the background. Instead of trying to stain the cell itself, you would stain the background. And by contrast, you would be able to see that cell. Therefore, what dye would you use? Therefore, you use a negative dye. Why? Because the inside of the cell is already negative. You use also a negative dye. Therefore, because of that now, yes, they are going to repel each other. Therefore, the dye is going to stay in the outside. It's not going to penetrate that cell. But when it stays in the outside, it's going to take the shape of the outside of that cell. Therefore, here we go. You will be able to visualize that cell. Therefore, that's what we call negative staining. Yes, yeah, you can do negative staining for a lot of other reasons. But one of the obvious reasons why you would do negative staining, most likely, it's because the cell is too tiny. For example, Micrococcus luteus. Even though Micrococcus luteus is a gram-positive organism because we are going to do gram staining, but it is so tiny that you may have to do negative staining to be able to know, oh, those are micrococci or micrococci. Very, very, very important. Treponema pallidum, the agent for syphilis, Vibrio cholera for cholera. Okay? Very, very important. The agent for Lyme disease. Okay? Very, very important. Okay? They may be, very, 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 very tiny. Very, very, very tiny. Therefore, you may have to use negative staining to be able to see them. Or you could have to use some kind of, illumination. Therefore, you would use dark field illumination that can give you almost the same result as negative staining because they are very, 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 very tiny organisms. In fact, Mycoplasma pneumoniae, Streptococcus pneumoniae, Klebsiella pneumoniae, all of those other organisms can give, some kind of bacterial, can cause some bacterial pneumonia. Mycoplasma pneumoniae, because it doesn't have that kind of peptidoglycan cell wall. It is very, very, very tiny. It is this kind of bleb of protoplasm. Okay. Therefore, it may be that you cannot use gram staining unless you have some very, 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 very special stains. It may be that because it is so tiny, you may have to use some negative staining. Therefore, basic dye, they would penetrate the inside of an organism. Negative dye, acidic dye, they are going to stay in the outside, because they are negative, and most likely they are going to be repelled by the negativity of the cell wall membrane of an organism. Examples of basic dye, crystal violet, methylene blue. Okay. Very, very, very important. Safranin, amyloid. Okay. Examples of negative dye or acidic dye, for example, India ink, niacin, acid carbonyl fusion. We will talk about them. Then to distinguish an organism, to distinguish the shape, the arrangement, we may have to do a simple staining. Simple staining means using only one stain. That's it. Only one stain. Any questions?
No.
Mm-hmm. Any questions?
No.
Okay.
Yeah, I don't have
Chapter 1, Chapter 2, Chapter 3, Chapter 4, it's going to be in your first exam. Chapter 1,
Thank
2,
you.
3, 4. 1, 2, 3, 4. Uno, dos, tres y cuatro. 1, 2, 3 et 4. Okay, they are going to be in in your first exam. Okay. Now,
Professor, do we have dates for those exams yet?
I never have dates in my class. The only date that I really have, it's the date posted by the powers that be for your final exam. Because when there is date, I can give you an idea about dates. For example, I put a date to have the first exam. That means that if it's for exam, you don't understand this, there is no time for me to take my time and teach you and doing this. And then here we go. We need to do those four. Therefore, I am reading from a PowerPoint. I'm not, responding to questions to make sure that you understand, because I am a man on a mission. And then we miss the purpose. I know that some people are about dates. I'm not about it. If I have to put dates, that's what you want. We are going to put dates, and we are going to go by the dates, and we will not change it.
Okay, so what I'm understanding is you like to go at whatever pace you're seeing is helpful for the class to get through the material so that we're all learning.
Yes.
Okay.
Yes, that's the way that I do it. I
Okay.
do not put dates. You understand? Because so there are some chapters, they are very challenging here. You understand? If I put dates, I have to go by the dates. I have the exams, it's already written, and you don't understand, you have a lot of pressure, then that defeats the purpose. My purpose is for you to understand what you're doing. There is no point of giving exams when you don't understand. I know people want to have, oh my God, I need to have dates. All that you need to do, go and do 1, 2, 3, 4, and you will be ready for whenever that we have the exam. And come with questions for me.
Okay.
Does that make sense?
Yes. So just for in terms of being prepared for class, should we assume like one chapter for each lecture or for every 2 lectures?
Let's say 2 lectures.
Okay.
You see? Yeah. Okay. 2 lectures. Because the problem is, it's not all of you that are at the same level. Some of you haven't taken biochem. We are going to hit hard on some stuff that are like from biochem. And some people are going to raise their hand like every 2 minutes. And you're going to be like, oh my God, you understand? Yeah?
Yeah.
Okay. And it may be that we have to take 2 lectures for 1 chapter. There are some chapters that are challenging.
Right.
You understand?
Yeah, totally understand. I've never had a professor who does that, but I really appreciate that because you're able to make sure that we're all learning. And we
Oh yeah. Okay.
Yes, thank you. Thank you.
Any other questions? Anything else?
Nope.
Okay.
No, thank you so much.
Okay. Therefore, if we see here in staining, this is an unstained specimen, and this is a stained specimen. Sorry. Okay. And how do we do this? Now, I'm going to explain this because we are going to do it again in lab. Okay. First of all, we need to prepare a smear. Okay. We need to prepare a smear. What does that mean that we prepare a smear? Okay. Yeah. That means we are going to do you remember that I gave you the 5 I's today? What was the first I?
Incubation.
We added inoculation, isolation, investigation. The first one.
The first one was inoculation, right?
But yeah, inoculation.
Okay. Yeah. That means here, if you remember also today, what we did, we took, a bacterium, you understand, from point A, and we transferred it to point B, right? Therefore, we took a bacterium from a tube or wherever, a sampling, and we're going to put it on a slide. When we put it on a slide, most likely, yeah, we are going to spread it over the slide according to a certain diameter. You understand? Yeah. And we are going to let it dry. Okay. We can let it dry in the air. When it is dried, we want to make sure, okay, that whatever that we put here is going to stick to the slide and it is not alive, because we're not doing a live stuff. Okay. Therefore, we are going to pass it on top of fire. Okay. We do it so that we can fix it, okay, into the slide. And then now we're ready for staining. We are going to stain it. Okay. When we stain it, it may be that we are doing a simple staining, which is using only one stain. What do we want to do? We want to achieve with simple staining all that we want to achieve. Yeah, it's to distinguish the organisms, their shape, their arrangement, their size, shape, shape, size, and arrangement. And then, of course, we use a microscope to actually to be able to see them. Very, very, very important. And of course, if we want to see more characteristics, we want to go to higher magnification, you understand? And of course, we might have to use some kind of oil, immersion. But then we might have to do what we call differential staining. Therefore, what is a differential staining? Differential staining, most likely, this is a stain that could help you, okay, to distinguish the different characteristics, certain characteristics from that cell. Therefore, we need to use, okay, a differential staining technique. Most likely, when we use differential staining, we will be using at least 2 stains. At least what? 2 stains. Most likely, of course, we are going to see the cell's shape, the arrangement, color. You understand? Yeah? Okay? But it may be that we're looking for some other types of characteristics. Do they have a cell wall? Okay? Do they have a big cell wall? Do they have a small cell wall? What do they have inside of them? Do they have ribosomes? What do they have? Now we're looking for some other types of, okay, yeah, characteristics. Examples of differential staining, gram staining, okay? Yeah, okay. Why would we perform a gram staining? Well, it can give us some very, 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 very valuable information, okay? Most likely. Yeah, for example, we're using it in medical parlance, because it would help us to design a best fit antibiotic so that we can actually neutralize those culprits. Gram staining. Okay, we want to know their cell wall. Does it have a lot of peptidoglycan that we are going to talk about? And then we can use penicillin. Yeah, because it has a lot of peptidoglycan. Because yes, we have designed an antibiotic, or we have seen there is an antibiotic that can destroy the peptidoglycan cell wall. Because the cell wall gives structure. The cell wall gives structure. The cell wall gives structure to the bacterium. Therefore, if we destroy the peptidoglycan, therefore, most likely, yeah, that bacterium is going to lose its structure, and whatever that it has inside is going to spill out. Therefore, we're killing that bacterium. Very, very, very important. Okay. Now, please, I'm going to stop here because this class ends at what time? 8:10, right?
Yes, 8:10.
Yes.
Okay. Because we had only a 5-minute break, we're supposed to have kind of like a 15-minute break. I wanted to do that. Okay. Please remind me next time. Write it down, please. Write it down. Write it down. The plan is to do gram staining. Okay. And then I am going to give you something that you should never, never, never forget, okay, from your medical, okay, from your medical endeavors later on. Never. You should have it right here. I wake you up at 4 o'clock in the morning and I ask you that question. Those questions you should be able to answer. Therefore, write it down. Next time, that's what we are going to cover. Very, very, very important. In fact, I am going to try to cover them also in the lab. Please remind me when we're doing gram staining. You understand? Okay, because I need to teach you that stuff. It's very, very important so that you can actually use it, okay, to your advantage. Okay, medically important bacteria, gram positive, gram negative. Prepare yourselves accordingly, people. Okay, any questions? I'm going to put some of the homework. You know, they are already available. It's the dates. That's why you don't see them. The dates are a little bit off. That's why you don't see them. I'm going to correct the dates. Any questions, please? Any questions?
No questions, Professor.
Okay. Therefore, thank you very much. See you, please, next week. Okay. See you next week. Okay.
Have a great evening, Professor.
Yes.
Have a great afternoon.
Thank
Okay.
you, Professor.
Thank you. Okay. Bye.